Hi everyone, my name is Aditya and today we will be examining plant reproduction. We'll be looking at some of the structures involved in plant reproduction as well as the processes behind reproduction, growth and development as well. Okay, so firstly let's examine 9.3.1 of the IB syllabus, which is having a look at some of the key reproductive structures and organs involved in plant reproduction. In this diagram, we can see a dicotyledonous animal pollinated flower, otherwise known as an angiospermophyte, with several reproductive structures. So firstly, the sepal down below, and these are an outer ring of small green leaves, which enclose the flower in a bud and help protect the bud as the flower undergoes early development. So then we have the petals, collectively called the corolla. These are colored surfaces which are bright pigments to attract pollinators like insects, birds, and bats. So the flower itself can be considered hermaphrodite, as it has both a male and female reproductive part. The male reproductive part consists of an anther, which is structures which produce haploid pollen by meiosis and house these pollen grains. And the anther is supported on top of stalks known as filaments. So the female part of the flower consists of carpels, which in turn consists of the ovary, which is a structure which contains the ovules, and this is where fertilization occurs between the male and the female gamete. And the other parts of the carpel are the stigma, which is a sticky surface where the pollen lands and is activated, as well as the style, which is basically a connecting structure. So hopefully you can see all these structures clearly labeled on this diagram. So as we continue with our examination, under the IB syllabus 9.3.2, it's important to distinguish between three key terms, which are pollination, fertilization, and seed dispersal. So the diagram right here shows the process of pollination, which is defined as the transfer of pollen from a mature anther to a receptive stigma. So in some cases the pollen may come from the anthers of the exact same flower or different flowers within the same plant, and this is known as self-pollination. But the more common mode of pollination is where pollen is transferred from different plants of the same species through either the wind or pollinators such as birds, bats, and insects, which are attracted to the flowers by a sweet sugar solution known as nectar. So this type of pollination is called cross-pollination, and is seen in the picture here with the hummingbird and the bumblebee acting as pollinators. So by contrast, what this diagram shows is fertilization and seed formation and dispersal. So fertilization is the fusion of the male and female gametes to form a zygote. So in this diagram, what we can see is that once the pollen is dispersed onto the stigma, it produces pollen tubes which grow down through the style using digestive enzymes until they enter into the ovule through an opening known as the microbial. So two male nuclei enter through the pollen tube. So one of the nuclei of the haploid pollen nucleus fuses with the egg nucleus to form a diploid zygote. And what's really unique about plants is that the other pollen nucleus fuses with another nucleus to trigger the formation of the food store for the embryo. And this process is known as double fertilization, and we'll be having a more in-depth look at it in a bit. So finally, what we see this bird doing here is seed dispersal, which is the carrying away of the seed from the vicinity of the parent plant. So while several plants do have structures which have evolved to enable seed dispersal by wind or water current, for example, the kernels in wheat plants are light and they enable seeds to be easily dispersed by the wind. But a lot of plants do rely on animals, birds, or insects to distribute seeds in order to enable the plants to grow and reproduce in different places. And this obviously decreases the level of intra-specific competition each individual plant faces. So some plants even rely on an explosive mechanism where the seeds are flung away from ripening fruit. And most seeds are obviously compact and lightweight to enable such dispersion and seed dispersal easily. So now we'll have an in-depth look at syllabus detail 9.3.3, which is the internal and external structures of a typical dicot seed. Okay, so now we'll move on to IB syllabus detail 9.3.4, which is the conditions required for the germination of seeds. Okay, so some of these probably seem obvious. While a good amount of water is needed to keep the embryo fully hydrated and maintain hydrolytic pressure. So there must also be enough partial pressure of oxygen to sustain aerobic respiration of the embryo, because remember the embryo doesn't photosynthesize as yet. And finally, there must be a suitable temperature close to the optimal temperature of enzymes to catalyze metabolic reactions. So this range is typically between 1 to 50 degrees, but it really does vary a lot from plant to plant. So now that we know the conditions needed for seed germination, let's have a look at the biochemistry and the steps of seed germination, which fall under IB syllabus detail 9.3.5, which is outline the metabolic processes during the germination of a starchy seed. So the first step of the process is that with adequate temperature and oxygen, water is absorbed, and the hydrolytic pressure causes the tester to split. So after this, embryo cells begin to produce gibberellic acid or GA, which is a growth regulator. The GA then passes to the food stored in the cotyledons, where protein reserves are converted into hydrolytic enzymes, especially amylase, which in turn hydrolyze stored starch into maltose and then glucose. And proteins are also hydrolyzed into amino acids, both of which sustain respiration and provide building blocks for the synthesis of intermediates for new cells. So once glucose and amino acids are translocated to growing points of the stem and root, these stimulate growth and development there, and eventually a self-sufficient autotrophic seedling emerges. So finally, let's see how the flowering of these plants are controlled. And this falls under 9.3.6, which basically explains how long day and short day plants and the flowering are controlled, including the role of the phytochrome. So obviously different plants flower at different times of year. So when flowers are produced and not produced for the rest of the year. So daylight provides really important signals, and a blue-green pigment called phytochrome is involved in the flowering process. So phytochrome is present in most green plants in small concentrations. It's a large conjugated protein consisting of the protein and pigment molecules combined, and it's highly reactive and able to absorb different wavelengths of light. So there are two types of phytochrome: PR, which is blue and absorbs red light of wavelengths of around 660 nanometers, and PFR, which is blue-green and absorbs far red wavelengths of around 730 nanometers. So when PR is exposed to the light or red light, it's converted to PFR, which is converted back to PR when it is exposed to dark light or far red light, slowly and quickly respectively. So the entire process is known as photomorphogenesis, where plant growth is influenced by light, and photoperiodism is the response of an organism to changing lengths of day. So it's mainly PFR which controls the onset of flowering, and flowering plants are divided into two categories. Short day plants, which only flower if the period of darkness is above a certain critical length, because PFR inhibits flowering growth. But long nights faced by these plants often remove the inhibition by causing PFR levels to drop to a low level. So conversely, long day plants only flower if the period of uninterrupted darkness is longer than a critical day length, as PFR promotes flowering growth in these plants, and a long daylight causes PR to accumulate into PFR. So with that, we've come to the end of the presentation and the podcast, and I hope you now know more about plant reproduction. Thank you.